That actually gets me to my next topic, lipids. Okay, so we talked about DNA at the very beginning. We talked about protein structures, sort of, for almost entire 1/3 of the class. So for this week and maybe a little bit of next week, we're going to spend some time on lipids. Now, a couple things I do want you guys to know. Of course, understand and familiarize with the nomenclature of fatty acids. And so what do we call fatty acids? And there are going to be some systematic names for fatty acids. Also, understand the structures of lipids and their characteristics, and understand how lipids make you membranes. And then we'll talk about membrane-associated proteins, transmembrane proteins, and then not this lecture, but the next lecture, we'll have molecular transport across membranes via different mechanisms. Okay? So this is sort of what we're going to be discussing. So, occurrence of lipid in biology. Lipids are everywhere, especially in food. You know, we are evolved to like lipids, believe it or not. When you're hungry and you smell something very oily or fried, you feel like great. Okay, you want to eat it. That's because you're evolved to do that. So lipids are a broad group of molecules with the characteristics of having long hydrophobic carbon chains. Lipid molecules are commonly found in fats, oil, waxes, certain vitamins like A, D, and E, soaps, etc. Okay, so it's like almost everywhere. The most significant function of lipids for biology is the formation of membrane. I say this is the most significant function because it goes back to, you know, at the very beginning we discussed about Kochlin's 7 pillars of life. So be able to circle something and say, okay, this is inside and then whatever is outside. This, lipids in biology, is very useful for that. Draws the boundary of what's inside of the cell and then what's outside of the cell. Okay, you have a separation in space. Okay, separation in space, that's important. So, lipid molecules, what do they look like? You can have something like soap. You see all of these hydrocarbons, so they're all attached to hydrogens, right? And then essentially this is a fatty acid. Okay? So soaps is essentially fatty acids. Or you can have triacyl glycerides, TAG. This is triacylglyceride, you know, lipid storage. This is completely hydrophobic. Lipid storage, okay? When you go to the hospital and get your normal body exam, your body checkups, this is something you absolutely measure. 血脂肪, 三酸甘油酯. This is what this is. And then you have vitamin A, different forms of vitamin A. When you look at vitamin A, what you see is that with the exception of this is an alcohol, everything else, you see how it's all carbon and hydrogen. If it's all carbon and hydrogen, this thing is what? It's going to be hydrophobic. Why? Because you cannot do hydrogen bonds. Okay? No hydrogen bond, no polar. So all of these are lipid soluble. The molecular structure and behavior of lipids. Let's first ignore vitamin A and that kind of stuff. Let's first talk about lipids. Okay, so lipids in a sense that we're talking about fatty acids. Okay, we're just talking about fatty acids. Most lipid monomers in general will contain a hydrophilic head and a hydrophobic tail. And typically a lipid molecule is insoluble in water, even if it has that hydrophilic head. It is usually insoluble in water. Okay? For example, I can tell you the story of my student a couple days ago in my lab trying to create a solution of hexanoic acid. Okay? Hexanoic acid is 6 carbon and then acid at the end. Okay? And you think that's not too bad, right? It's only 6 carbon acid. All right? believe it or not, okay, that thing is not very soluble. It is soluble with water, okay, to maybe a small concentration. It will not be entirely soluble. So what my student did was he poured hexanoic acid, added water, very beautiful. You see two layers, organic layer and an aqueous layer. Okay, so it's actually something as small as maybe 6 carbons. It's already solubility become very, very low. Okay, so imagine fatty acids, if you have maybe 16 carbons, it's going to be very insoluble. Now, having said that, however, okay, lipids can associate with each other and form water-soluble structures. Okay, so what this means is that by themselves, even though they are insoluble, but if they are in low enough concentration, so the concentration is low enough, these lipids will actually start to aggregate and then form soluble structures. They are not soluble by themselves, but then they can arrange into a 3D structure that makes it soluble. These 3D structures are micelles, vesicles, bilayers. How do they do that? Essentially by displaying the hydrophilic head on the outside. So you'll have hydrophilic heads on the outside, and then hydrophobic inside. So outside is water, inside is essentially oil phase. Then no problem. It becomes soluble as a whole. But individually, they are not very soluble. So no, when lipids are used to make energy storage, they are turned into completely insoluble oil. So triacyl glycerides, so TAG, this thing, if you look at this structure very carefully, you do not have a hydrophilic head. There's no hydrophilic head. You look at, where's the head? Maybe this is the head. If you look at it, is there anything that will form you hydrogen bonds? No. Okay? Not significant at least. Right? This is CH, nothing there. This O has lone pair electrons. That's fine. You have lone pair electrons here, lone pair electrons here, but you don't have the protons to make hydrogen bond with. Okay, so this thing is essentially completely insoluble. Okay, and you know that how you go home and take some cooking oil, dump it in water, it's insoluble. Okay, right, makes sense. So if it's completely insoluble, then you'll have hydrophobic effect. Okay, so we're not going into detail about hydrophobic effect. You should remember what this is from our discussion long time ago. Recall that this is an entropic mechanism. Okay, so if you have insoluble stuff, all the insoluble stuff will come together, kick out the water, because you want to minimize water-lipid interface. So as I was saying, even though these lipids by themselves are not soluble inside water, but when you have them in low enough concentration, they actually form soluble structures. And we have micelle is the first example. Oh yeah, by the way, all of these structures are quite useful for gene therapy, like gene delivery, protein, like drug delivery. Okay, they're actually used a lot. So there are companies that make money by essentially developing methods, and especially using these lipotransporters that can encapsulate different drugs inside of those things and then try to have them as a carrier to deliver this drug to whatever cell. So this is actually very relevant to what we're studying. So the first example we have is micelle. Micelle is like this. It's very simple. You have essentially two layers. One is the outside layer that's going to interact with water. So outside is aqueous, inside is organic. Okay, because inside, as you can see, this is the oil phase. Okay, so it's hydrophobic on the inside, hydrophilic on the outside. Okay, this is micelle. Now, micelle is different from vesicles. Okay, vesicles, you will have 3 layers. You'll have outside layer being aqueous, so that's the water outside. You have a middle layer of organic, of oil inside, and then you have another layer of water, essentially aqueous interior on the inside. Okay, so if you look at the pictures carefully, what you see is that I zoom out this, then you have a head, two tail, and then on the other side of the two tail, you have another two tail and a head. Okay, so vesicles are essentially smaller version of lipid bilayer. Okay, so lipid bilayer is just an extended version of vesicle. Okay, so understanding that your lipid bilayer, it will never terminate like this. It will always, okay, essentially circulate around and then form a larger enclosure. But lipid bilayer and vesicle, they're pretty much the same thing. You have a outside is going to have water interface, and then you have a lipid oil phase. That's sort of the membrane itself. This is the membrane itself. And then you have an inside that's also water soluble. Okay, so having vesicle or bilayers, that is the prerequisite for life. Okay, so this is what achieves the spatial separation. You have outside water and then inside water, because all enzymes, they function in the presence of water. So inside of your cell is also water. So it's not oil, right? So you cannot have micelles for membranes, for cell membrane. You have to have vesicle or bilayer. And vesicle and bilayer, they're pretty much the same thing. It's just that vesicle is smaller. Now, micelle, on the other hand, sometimes this thing, micelle, is very useful for delivering hydrophobic stuff, right? Because inside you have a hydrophobic layer. And then you can throw in it maybe hydrophobic drugs, hydrophobic molecules that you need to deliver. So micelles are still very, very useful. Regardless of micelle or vesicle, if we look at one subunit, you have, this is going to be something like glycerol, and then you have two lipid arms. And what makes this monomer in building the vesicle or micelle is this one small subunit called the fatty acid. So this is the fatty acid. And fatty acid is sort of the simplest lipid monomer. Fatty acids can make up other more complex lipids such as triacylglycerides. Fatty acids are essentially long chain or long carbon chain carboxylic acids, typically with even number carbons. Okay, so we'll talk about why it's always essentially, most frequently, it's even number. So next semester we'll talk about why it's usually always even number carbons. Okay, it has to do with its biosynthesis. The way that it's synthesized will always give you two, its multiples of 2. Fatty acids can be saturated, or unsaturated. If it's saturated, it only contains carbon-carbon single bond. If it's unsaturated, it means that you have double bonds in them. So this is in Chinese what we refer to as 饱和性脂肪 and the 非饱和性脂肪. So in nature, unsaturated fatty acids are mainly the cis fatty acids, cis fats. You know from organic chemistry, alkenes, you can have two stereoisomers. One is the Z form and one is the E form. Remember that? Essentially, they're sort of, your carbons, do they point the same direction or the opposite direction? Fatty acids, if it's unsaturated fatty acids, usually you will always see the cis fatty acids. So if you look at it, so we're going to take a look, it's always going to be like that, not like that. This is usually synthetic. Trans fatty acids are usually synthetically produced as a side-contaminating product of food industry. And consumption of these fatty acids will increase the risk to heart diseases. Okay. Do you guys actually, do you know where we get these trans fats? Right, so what's the original, 你知道它那个原始的 substrate 是什么? The original fat that you use to make this butter replacement. Butter is animal fat, but if you want to use the vegetarian butter, it's synthetically derived, right? So it's actually made by hydrogenation of vegetable oil. Vegetable oil and then you have a hydrogenation to do that. Now, if you do a synthetic hydrogenation, you're going to have non-specific or dehydrogenation. Yeah. So in that process, you will get some amount of trans fat. And as a result, those things are not natural. And if it's not natural, your body doesn't really know how to degrade it. And then as a result, it's usually going to accumulate and then do some funny stuff to your metabolism. Okay, so that's not good. The natural version of unsaturated fatty acids is usually cis fats. A saturated fatty acid, of course, every bond is a sigma bond, right? Carbon-carbon single bond. So they can all freely rotate. So essentially they can rotate, right? The entire thing can rotate very well. But usually we just say, as an average, the most stable configuration or the conformation, it's going to be a linear, sort of a linear chain, which is why when we draw lipids, we always just draw it like that. Sort of, we consider it as linear. Now, if you have unsaturated fatty acid, you will have a little kink. So you'll have like that, and then all of a sudden you have restriction in the rotation of this particular bond, right? If you have an alkene there, you cannot freely rotate. If you can't freely rotate, you set the direction. And then here you can rotate again. So as a result, you'll have a little bend. Okay, you have a little bend in the molecular structure. Unsaturated fatty acids. Now, humans, for us, when we make our lipids, we primarily make saturated fatty acids. So most animals, we primarily make saturated fatty acids. We can make unsaturated fatty acids, but limited type. So here it says that humans can make saturated fatty acids and most other fatty acids needed by our body by metabolizing carbohydrates and proteins. Okay, so we can take glucose and then convert that into fatty acids. While we can make saturated and most other ones that we need, but we cannot make omega-3 and omega-6 fatty acids. And omega-3 and omega-6, they are required to synthesize hormone-like molecule called eicosanoids. These eicosanoids are C20 molecules. But we'll not talk about eicosanoids at this moment, but just for your information, eicosanoids are regulating compounds that are used to regulate, blood clotting, blood pressure, immunofunctions, and many different cellular processes inside of your cell. So they are primarily signaling compounds. Obviously, you sitting here thinking, that's probably pretty important. Why is that? Because if you don't have an adequate amount of these eicosanoids, then your body regulation will be messed up. If your body regulation is messed up, you're going to have different stuff coming out. Okay? That would be bad. Eicosanoids, again, produced from omega-3 and omega-6. Now, let's talk about what are omega-3 and omega-6. So let's say we have a saturated fatty acid. It looks like that. Okay? And we know that where do we start labeling? We start labeling here because this is the acid functional group, right? So this is C1, C2, 3, 4, 5, 6, blah, blah, blah. That's the normal numbering. Omega being the last letter in the Greek alphabet. Correct me if I'm wrong, but I think I'm right. It's the last alphabet in the Greek alphabet. So it means from the back. Okay, so if it's omega-9, then you have, this is omega. It's a W looking thing, just omega, okay, omega. Omega 1, 2, 3, 4, 5, 6, 7, 8, 9. And then this 9 is from 1, 2, 3, 4, 5, 6, 7, 8, 9. I should say omega, omega. Okay? So this particular carbon right here, that's the omega-9. Omega-9 we can make, no problem. Omega-6, meaning you go 1, 2, 3, 4, 5, 6. That's omega-6. Now, what do you see? Omega-6 typically will always have omega-9 as well. Okay, so when you have omega-6 oil, it's going to have 2 alkenes. Omega-3 oil, it will have 3 alkenes because you have 1, 2, 3. Okay, so omega-3, you'll have 3 alkene units inside them. Okay, so this is why you'll have polyunsaturated lipids. This will be called mono unsaturated, and then diunsaturated, and usually we just say polyunsaturated. You have more than one. So omega-3, you go from, at the end, count 1, 2, 3, first alkene, and then one space, so alkene, and then a space, and then another alkene. Okay? So you have a little space right there. So this is the distribution of different fatty acids in common cooking oil. So again, you have saturated, monosaturated, and then you have the 2 polyunsaturated. Okay, so as you can see, you have different kinds of cooking oil, and most of them are vegetable oil other than chicken fat, pork fat, beef, butter. So these guys are animal. And what you see is that in animal oil, primarily we see saturated and monounsaturated, right? Because saturated we can make, so animals like we can also make that, and then monounsaturated is referring to omega-9, we can also make that, right? So those things that just like us, we can make it, no problem. Now, if you look at canola oil or sunflower oil, olive oil, so all of these things, these are all vegetable oils on top. So vegetable oils typically will have more degrees of unsaturation. And so, which is why people always say, oh, you know, eating food made with olive oil is good for your health. Okay, the reason is because it provides you the needed omega-3, omega-6 oil that you need to provide a healthy balance in regulation. Coconut oil, if you look at coconut oil, it's pretty crazy, even though it's a vegetable oil, but it's primarily saturated. Now, knowing this chart, okay, immediately you can draw a little conclusion here. Butter and coconut oil, it's a solid or liquid. So primarily it's solid at room temperature, right? Coconut oil, same thing, right? Coconut oil, like coconut, I slice it in half, and then 那个椰子水就是拿出来以后，里面那层白色那些东西有没有那个果肉 ？That's coconut oil. Okay, 那其实都是lipids. Okay, and that's solid, right? That's solid. You don't have a choice. Okay, those things are solid. So what does that tell you about your fatty acids? If you look at butter, coconut oil, those things are solid. And if you look at their distribution here, immediately you see that they are primarily saturated or monounsaturated, right? So what that tells you is the more saturated fats you have, the more likely it's going to become solid at room temperature. Okay, we're going to have this crazy systematic way of representing fatty acid. So X, Y, parentheses, C/T, delta, N, you're just like, what? Okay, now the X here refers to the number of carbons in total length. So if we look at this example, we have 18,3, so it's total of 18 carbons. 3C, meaning 3 unsaturated bonds. C, meaning cis unsaturation. And then delta, 9, 12, and 15 are the position of the double bond location. Okay, so you have X, Y, and then whether it's cis bond or trans bond, and then the location. Okay, so that looks like this. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, right? And we have 3 double bonds. 1, 2, 3. Location, 9, 12, 15. Okay, so that tells you immediately what is the structure of this particular fatty acid. Now, this is the nasty part. Okay, for every fatty acid, we're going to have a common name. It's nasty. Now, you're going to ask me, do I have to memorize this? Like I said, my answer to all this type of question, okay? Do you have to memorize this? No, you don't have to memorize this. But if you are going to be a good biochemist, I know you don't want to be and you don't really care, okay? You should know, okay? This is something that I think you should know. You know, when I was an undergrad student, my professor made me memorize this, and I did. You know, after I take the exam, I immediately forget all of them. That's going to happen to you. Okay, let's be honest. Okay, let's be honest. That will happen to you. But you should at least be able to recognize their systematic names. Systematic names are very, very easy. It's like, I don't know, tri, tetra, penta, blah, 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 right? If it's 10, it's called deca, like decanoic acid. If it's 2, it's dodecanoic acid. And then 14 is tetradecanoic acid, hexa, octa. Those you should be able to know. But the common names, if I ask you a question about fatty acids, I will give you the common name. I will give you the systematic name. So you should at least know the systematic names. Now, pay attention. Here we have saturated fatty acids. When you go down to the unsaturated fatty acids, you will have, notice here is blah, 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 anoic acid, or hexanoic or decanoic, right? It's AN, indicating that it's primarily made out of alkanes. Here is enoic acid, so it's talking about alkenes. Okay, just make sure you remember that. I think you probably heard about this back in organic chemistry, but you know, this systematic names, please, you should at least know. The common names are useful. They're really, really useful. I'm not even kidding how useful these things are. It's useful. But it's not essential that you absolutely remember, at least for my purpose. Right? But you should recognize some of these, because if you know, let's say if you go on a trip to the United States or whatever, and you want to buy potato chips, 你买一个东西嘛，食物相关的，然后你看后面那个 ingredients, okay, and you look at the ingredients and you see linolenic acid, and you just like, I wish I paid more attention to biochemistry back in college, right? I mean, it would be great if you immediately know, okay, this thing has double bonds, okay? I don't know what that would be useful for, but at least you know. You can show off to your friends and say, hey, I know what it is. Now, some fatty acids, sometimes they will have branching and even cyclic. So these are the weirdos, right? They're really weird. You don't see them a lot, so we're not going to worry too much about this. On this chart, there's another very, very useful information. That is the melting point. As you can see, first of all, what is the melting point? The temperature which you change from solid to liquid, right? That's the melting point. So essentially, melting point decreases as you go from long chain to short chain. So you have 31.6 for C10, 88.5 for C26. This is all saturated, straight chain fatty acid. C10, melting point is 32. For C26, melting point is 88.5. So we have a decreasing in melting temperature as a function of a decreasing number of chain inside this molecule. Why is that? Because the more chain you have, the longer the molecule, the more what you have. The more van der Waals interaction you'll have between the molecule. As a result, they pack better. Because the longer chain, the longer carbon you have, the more contact surface they have between the hydrophobic parts. So then they pack really, really well. So then you will have a higher melting temperature, right? They pack better. Now, if you look at the unsaturated, let's look at C20. C20 here. So if it's saturated, you have 76.5. If it's C20 with 4 alkenes, you have minus 50. That's a huge 120 degrees Celsius difference in melting temperature. Why is that? Because when you have alkenes, remember we say every time you have an alkene, you have a fixed angle for the lipid arm. So the more alkenes you have, your lipid essentially will have awkward spaces, meaning it's going to be twisted like that, twisted like that, twisted like that. Okay? As a result, the packing sucks. So if the packing is not good, then it's easier to become liquid. This has a significant function for different biological systems at different climate. Okay? For example, if you live in a very cold environment, your membrane is not going to be made out of saturated carbons. Why? Your membrane will be freaking solid. That doesn't make sense. Can you imagine if your membrane is solid, like your cell is a big chunk of solid? It doesn't work. No, it doesn't work. Okay? So this is why, what kind of material has a lot of omega-3? Fish oil, right? And usually fish oil, these are deep sea fishes like salmon, right? Salmon has a lot of fish oil. Fishes, they grow in hot places or cold places. Probably not going to be like very hot places, right? Like water usually is colder than, the atmosphere, okay? Especially if it's deep sea fish. But as a result, those fishes usually will have a larger percentage of unsaturated lipids, because if they don't, they live in a very cold environment. If their lipids are made out of saturated fats, it's going to be a solid, and solids are not going to be good for any organism. This has a significant function or role in different organisms' membrane. So a specific type of fat we have is triacylglycerides. Triacylglycerides, again, it's completely insoluble. Okay? This is a fat storage, or rather energy storage. So this is essentially fats or oil. So triacylglycerides, also known as, so it has many names, by the way. Yeah, it's also known as triacylglycerol or triglycerides, TAG, whatever. I usually call them triacylglycerides. It's an ester of glycerol plus 3 fatty acids. Now, these 3 fatty acids can be saturated, unsaturated, whatever. It depends on the type of organism you're dealing with. If it's plant oil, like olive oil, it's going to have a lot of unsaturation. 那个橄榄油的话，就是会有很多 alkene 在里面。所以 this comes back to my question of why is vegetable oil primarily in an oil state, in a liquid state? It's because they have a lot of these unsaturation, and then these unsaturation will lower the melting point. Glycerol, this is what this thing is, glycerol, okay? And glycerol is a 3-carbon molecule that has hydroxyl group on each of the 3 carbon. And essentially, when you are making triacylglycerides, you will take H here, couple with the OH here, so you get rid of the water, and then you link them together and you form esters. So just as with sugars, in order for this reaction to occur, your glycerol and fatty acid have to be activated. But of course, we will not get to this semester. We'll talk about lipid synthesis next semester when we talk about how you actually make these things. This is something that I want you guys to always remember. In biochemistry, organic acids, so basically carboxylic acids, is usually very, very stable. So if you want to do reaction with carboxylic acids, you have to have some kind of activation, okay? Either to the fatty acid, the organic acid itself, or something that reacts with it. Acids are just way too stable for any reaction inside of our normal temperature and pressure. Organic acid, too stable. Why? Resonance, okay? It has resonance, so it's very stable. Note the orange hydrophilic head is gone after binding with glycerol, so you don't have no more hydrophilic property. So TAG, triacylglyceride, they're insoluble. So inside of your body, you have these things called the adipocytes. adipocytes. You guys will learn more about blah blah whatever sites when you go to like cell biology or physiology. You'll learn all sorts of sites. I don't even know what kind of sites you have, but I know adipocytes is talking about fat cells, okay, cells that produce a lot of lipids. So you got a lot of these adipocytes inside of your adipose tissue. So these are fats. So they will form, they're actually inside of your body, they form these insoluble oil droplets. Yeah, it is oil droplet because even though we say the melting temperature is it's really, really high, but you know, for most part inside of your body at body temperature, you don't have like insoluble chunks, okay? But there's sort of like little fat globs inside of your cell, okay? Fats, you have two types, right? You have the brown fat and the white fat, okay? The energy containing or the energy storing ones are the white fat cells. So if you like to eat, I don't know, when you go and have a hot pot, you have a slice of beef. You see that white stuff? That's the adipocytes, okay? The adipose tissues. Functions of fat storage in animals, you have several functions. You have energy production. So this is one of the primary functions for animals. Most animals is oxidize for the generation of ATP. This we'll talk about next semester. We'll talk about how you degrade fatty acids. Now, the bad news is fats cannot make sugars, okay? In humans. We cannot make sugars from lipids. What does that mean? That means that if you gain fat, like lipids, it's very difficult to lose them, okay? You don't get sugars from lipids. You have to like exercise to hell and then you burn the fats. You have to like literally burn the fats. Lipids has to go, if you want to lose lipid, you have to burn it into CO2. That's the bad news, right? So you can't just convert fat into sugar. Bacteria can do it, we can't. You can also use lipids for heat production. There are specialized cells. Usually they're called brown fats. They will oxidize triacylglycerides and they will generate heat. So you obviously understand we can live in an environment of maybe 15 degrees C. Even though if your body is at 15 degrees C, you're dead. So obviously your cell or at least your body has to be able to generate heat somehow to maintain your 37 degrees. Insulation. Insulation is to prevent heat from losing inside your body. You guys know that lipids, they're not very good thermal conductor. So having a layer of lipid on you really prevents heat loss. So if you have friends that are super buff, okay, like I have friends who like work out every day and they're super buff, right? You would think that, you know, they're very macho and like very manly, but then actually during winter, they get cold very easily because they have primarily muscle cells on them. They don't have much lipid, and as a result, heat loss is very quick. So then they feel cold very rapidly. Right? So like people that have a lot of lipids on them usually can survive in winter time better. So again, not all triacylglycerides are the same. So again, this we talked about. Butter and vegetable oil, they're both fats. They're both triacylglycerides. But then for butter, you have them primarily in the solid state, and then this is primarily in the liquid state. And the reason for that is, of course, being the number of saturation, or the number of alkenes, or the degree of unsaturation you have in your lipid. That defines whether it's going to be a solid or if it's going to be a lipid. So again, you know, the thing that I talked about, the packing, is right here, down the bottom. Unsaturated fatty acids will increase the fluidity of the triacylglyceride. So it will decrease the packing of the different lipids. As a result, it increases the fluidity. As a result, you will primarily have liquid state for that kind of lipids.